Hi, my name is Arthur Mack. I joined the Chinese University of Hong Kong in 2011. My research interest is in tissue biomechanics, particularly the biomechanics of musculoskeletal tissues. Tissue biomechanics deals with the relations between tissue structures, tissue properties, and tissue function. How mechanical stresses are distributed inside the body tissues when they are loaded. How these tissue stresses relate to growth, development, aging, degeneration, regeneration, and remodeling. For many years, I am interested in deep tissues ulcers, when body tissues are subjected to moderate but prolonged mechanical stresses, and how to design body support interfaces to reduce the risk of such ulcers. Such ulcers arise from localized injury to the skin and or the underlying tissues, usually over a bony prominence, as a result of pressure or pressure in combination with shear. My research interest is motivated by the clinical challenge faced by many people with disabilities, like a person with spinal cord injury who needs to sit on a wheelchair for a long time, like a frail elderly person who needs to rest on bed most of the time. In recent years, my students and I have launched a series of multi-scale study of deep tissues injury under prolonged biomechanical and biochemical stresses. We assess the duration-dependent damage thresholds of muscle cells under mechanical stresses. Results support the hypothesis that chronic exposure to high-dosage oxidative stress could compromise the capability of muscle cells to withstand mechanical damages, while short exposure to low-dosage oxidative stress could enhance such capability. We examined the effects of oxidative stress on cytoskeletal actin polymerization and used an atomic force microscope to monitor the corresponding temporal changes in cell stiffness. Atomic force microscopy results shows that short exposures to oxidative stress apparently increase the stiffness of myotubes, but that long exposures make the cells softer. The turning point seemed to take place somewhere between 1 hour and 2 hours of oxidative exposure. We used femtosecond laser to evaluate how oxidative stress affected the pretension in the cytoskeletal fibers and the ability for cells to repair a micron-sized pore on the plasma membrane. We also find that mild but chronic oxidative exposure seems to have compromised the repair capacity of myoblasts when compared to the strong and acute exposure. We also find that intermittent vibration of 0.25 G at 35 Hz applied 10 minutes for every 30 minutes of compression could significantly alleviate the mechanical and oxidative damages caused during the 6 hours of static compression. We are currently incorporating our research findings into our design of a smart active cushion named VibroSag, with the objective to reduce the risk of deep tissue ulcers among people with disability.